Thank you all for coming. Lee Katz is the senior bioinformatician in the Enteric Diseases Laboratory branch at the Centers for Disease Control and Prevention. He graduated with a PhD in bioinformatics in 2010 from Georgia Tech and has worked at CDC since then. Since joining CDC, Lee has worked on several high-profile projects studying the evolutionary dynamics of the Haiti cholera outbreak and also aiding PulseNet, a national laboratory network that uses bacterial DNA fingerprints that make people sick to detect outbreaks, and using whole genome sequencing, or WGS, for genomic epidemiology. Lee has also worked on many software projects for public health, including the LiveSet SNP pipeline for genomic epidemiology, the Rapid Tree Making pipeline for WGS, also known as MASH-Tree, and the QA/QC system for bacterial WGS in public health, also known as SneakerNet. In his spare time, Lee, along with his co-host, produce a bioinformatics podcast called the MicroBinFeed Podcast, which can be found on many platforms such as Spotify and Apple Podcasts. Please help me welcome Lee Katz.
Thank you so much. Thank you for that warm introduction. I get to talk to you guys about a career in bioinformatics, and I just live down the street from you from Athens. Uh, and so hopefully this entices you guys to do something fun in bioinformatics, maybe public health, but um, I've had a really fun and great career so far. I'm just 10 or 15 years into it. I'm actually a native of Decatur, so I'm not far from where I grew up right now. I went on to Emory University to get a BS in biology and moved on to Georgia Tech to get a PhD in bioinformatics. Um, and then for the last many, many years, I've been a bioinformatician at CDC. Over that time, I've gotten three young kids with my wonderful wife, and yeah, just started picking up podcasting over the last 5 years, and I'll get into that at the end. And I really enjoyed my time on this track. I did my PhD at Georgia Tech. That's the Cherry Emerson building. My future wife at the time, and I took a helicopter ride over the building actually on a nice Groupon. And again, graduated in 2010 there. There was buzz. And then from 2010 till 2011, I was actually in the meningitis lab, and we did a lot of really interesting things, um, including including things like helping virtually eliminate meningitis A from Africa. In a large collaboration, I played a tiny, tiny, tiny part of that. And looking at the genomic basis for virulence in meningococcus. This is a group photo after uh, we uh, we received an award for our work in Africa for meningitis A. I'm over there wearing my sandals because I guess I just don't know how to dress sometimes. That's the future and now past acting director of CDC and Shuket over there. Really, really good times over there. There was a little bit of shift in my career. I moved over to the Enteric Diseases Laboratory branch in 2011. Um, I think this picture might be from 2015 or 2016. I don't know if Katie's actually in here. We spent several years together in EDLB, and actually now that I'm seeing this slide here with you, I'm embarrassed a little bit. I should have found a picture with us. We have a very large branch, do a lot of amazing things, and of course I treasure my time also working with Katie. This is the current team. So the previous picture was, I think, from 2015 or 2016. This is the current roster in my team, which is inside the branch from this year. My current team lead is Jess Chen. We have several people on the team divided into development operations on the left side here. I'm in applied bioinformatics and research on the right side here. And one of my students, actually, one of my coworkers is a student in from GSU, Alina Namira. She's a PhD student. I have a bunch of outside activities, so unfortunately, unfortunately, I'm a little bit scattered too. One of my outside activities from CDC is actually being an adjunct member at UGA, but on the Griffin campus with the Food Safety Informatics group. And so, if this becomes a very interesting thing, if foodborne bioinformatics becomes very interesting to you, UGA actually has a ton already. And if you want to go outside of um, like livestock and other things. So if you want to go into food, Griffin, Griffin, Georgia, the University of Georgia Griffin campus actually focuses on food itself, like lettuce and other things. I'm also a part-time instructor at Georgia State University. I teach the intro course for bioinformatics. So I'm going to get a little bit into CDC a little bit more. And while I go about this, I'll try to have pauses for questions, but um, feel free to write them in the chat or raise your hand, and we'll stop after a given slide to answer that. I like having a conversation rather just than just talking in one direction. Uh, so the Enteric Diseases Laboratory branch, this is where I am, and I guess this is about the time that I should make that disclaimer that all of the opinions I've expressed here and will express here are those of my own and not of my employer, not necessarily those of my employer. So, with that disclaimer in mind, I'm in the branch that houses PulseNet. I appreciate that Katie brought that up. This is the National Molecular Surveillance System for foodborne bacteria in the United States. It also has a very strong international component. Um, and I can answer questions about that later if you guys are curious. I'm not going to go into PulseNet International too much. In 1993, that was sort of the origin story for PulseNet. There was a very large outbreak in hamburgers at a popular fast food place, Jack in the Box, with over 700 illnesses. 4 children died. The individual states that had to address this started coming together and working together to see if they can run surveillance a little bit better on E. coli and other foodborne infections. In 1996, CDC sort of started taking the mantle and created PulseNet itself to help organize the states. Um, and this is the part where I sort of feel old because this is still about where you guys are either have been born or before you were born. So I'm feeling a little bit old here now, but I'm going to keep going on. Uh, zooming on through through the years, any outbreak that you might have seen in foodborne, uh, bacterial foodborne in the news, my branch probably has dealt with that. So in 2006, for example, there's a huge outbreak in spinach. 2009, maybe when you guys were starting to make memories, there was a huge outbreak in peanut butter, and maybe peanut butter was an important food for you when you were a kid. There's a huge outbreak for that. That's something that we had to analyze and study and help FDA get that off the shelves. In 2010, after the horrific earthquake in Haiti, um, we actually used that as one of the first examples where we started using whole genome sequencing to address an outbreak. And I can go into that in a little bit too. It was a really great use case. Again, awful tragedy, but scientifically, it was a good use case to observe an outbreak um, essentially on an island and observe the evolutionary dynamics of foodborne bacteria, or waterborne in this case. In 2013, we started turning our eyes to Listeria to see if we could run whole genome sequencing real time to see if we could actually switch our subtyping techniques over to whole genome sequencing using bioinformatics. In 2016, we had our 20-year anniversary, and 2019 and on, we started completely flipping the switch over to whole genome sequencing. I got my start in the foodborne branch with the Haiti cholera outbreak. And these are just a few different figures from my paper and a screenshot. So at the beginning of 2010, there was a horrible earthquake, and it's kind of well known that if you destroy or you have your water system destroyed, um, then foodborne and waterborne infections might start creeping in, and that turned out to be the case in October of 2010. We started observing a huge outbreak of cholera in Haiti, and we described the evolutionary dynamics of that outbreak. And I didn't bring too many slides for that because I just wanted to touch on a bunch of different things and give a good high-level overview. This is sort of where we got our start. We developed a SNP pipeline for this outbreak. We developed methods for showing these minimum spanning trees for these outbreaks and describing them a lot better. And honestly, internally, this helped us come up with better methods and better ways of communication internally with our epidemiologists and better ways to communicate it on slides and PowerPoint slides and reports to our epidemiologists and to our state partners. This helped refine a lot of the techniques. This turned into a pilot project later on in 2013. We thought, okay, we have the capability. Let's see if we can actually do this. Let's see if we can sequence all the listeria in the United States for a given year. We had just gotten our sequencer, and this was a very adventurous project. We made a huge collaboration with NCBI, with FDA, now the Human Foods Program after the reorg, and FSIS under USDA. So we all collaborated and decided to sequence all the listeria that we could find. And why did we do that? It's because of several reasons from several perspectives. The number one reason is that illness is rare but very serious, costly, and commonly outbreak associated. And believe it or not, every single case of listeria costs $1.8 million. This is in lost productivity and medical costs, medical salaries, medical everything. And it's huge. The current subtyping methods were not ideal. This is starting to become an old slide, I guess. So take that with a grain of salt. This is a slide from 2013, I believe. It had a strong epidemiologic surveillance component called the Listeria Initiative, and this is where our epidemiologists, they already were asking patients, like, what did you eat? What were you around? And they got an idea of what the commonalities were between the different cases. The Listeria Initiative was an epidemiologic um, uh, innovation where they had a recursive survey and they would ask, like, did you eat leafy greens? Yes. Okay. It seems like everyone's eating leafy greens. Let me go back to that patient and okay, did you eat kale? Did you eat spinach? Did you eat romaine? They could go into more details once they start figuring out what the signal is. And that had a huge impact. Also, on the FDA and USDA side, there's a strong regulatory component for listeria. If it's in food at all, if it's found in food, it is recalled. It cannot be sold. That's specifically for the species Listeria monocytogenes. And then for bioinformatics, the listeria genome is fairly small, stable, and relatively easy to sequence and analyze. Most changes in the genome are due to point mutations and not phages. That means that you can observe vertical evolution and get an idea of their relatedness instead of something convoluted through horizontal transmission. So looking at this, I borrowed a slide that my friend made on from a New York Times article actually in 2009. See if I can get rid of this Zoom menu bar so I can see the screen better. I think that did it. Whatever. Okay. So imagine you are eating food in the United States. Not hard to imagine. We do it every day. Uh, you get food from many different places. And in the case of some foods, like a hamburger, that food actually might come from several different places, get mixed in at a food processing plant, and then eventually when it gets to your plate, you've eaten a hamburger or something that's come from 10 different places in the world or in the United States. If you get sick from that, it's hard to trace it back to where it comes from and remove the contaminated product. So before 2013, we would use a technique called pulse gel electrophoresis, and that would give us something very similar to a restriction digest analysis on a gel. In fact, it's a very modified version of that. And so you'd see all the listeria cases in the US. The epidemiologist can ask, did you eat the hamburger? And we can look at the profile of the listeria that is found in their stool, and we can say, okay, this person and this person and this person in blue all have the same PFGE profile. They might be related to the case, to the outbreak. You can also get false positives to this method. Some profiles are more common than others, or you might just by chance have the same profile, so there might be a false positive here. You might also have false negatives. This person who did not test for that same PFGE profile might have actually eaten the hamburger. So there are false positives and false negatives with this. It's not perfect. And then how do we like really resolve this? Well, we can actually use WGS. We can use whole genome sequencing and resolve this. So let's blow that picture up, get a better resolution. So the question is, can WGS give us a better resolution? The answer is yes. And we've done this in many cases up to this point now. When this started off in 2013, we actually got an outbreak right away. In fact, we got this outbreak right before the official start date of it. So we hit the ground running on this with a multi-state outbreak of listeriosis linked to Crave Brothers cheese. It's a soft cheese that they were selling. And before I go into it too much, what I usually do on this is I assume that you guys, since you're all students, I assume that there's some lurker in here who just hasn't learned how to read a tree just yet, and that's okay. What I'm here to do on this slide is to just teach that without shaming and just, I don't even know who I'm teaching at this point. Somebody on this call just statistically doesn't know how to read a tree yet, and you should. So this is an educational moment for you. I'm slow walking that explanation so that that person woke up. So how to read a phylogeny before we talk about the phylogenies in the next slides. This is a tree. This is the direction of evolution on the tree. My text got muddled here, I guess, when I copied this one over. This is a taxon. This sample or this genome, now that we're in a tree that's called a taxon, this is a taxon, the plural. These are all taxa. This node right here, this dot right where they join up is the last hypothetical, last common ancestor, the LCA of taxa 3 through 7. We think it's hypothetical. We know it's hypothetical because we don't observe it. We observed the samples taxa 3 through 7. We never observed it, they're a common ancestor. So it's hypothetical. We also have numbers at these nodes for the hypothetical last common ancestors. These are percent confidence that we know they existed and that they group together these taxa. Good confidence here. And then we have the root. This is where like the last common ancestor of everything here. This is the scale bar. This tells you how much it mutates over a distance. Just like on a map, it tells you on a map like how many feet you've gone. This scale bar tells you how much evolution has happened over time. So 0.01, usually like percent of the genome. So 1% of the genome has evolved in this amount. On a tree, the vertical distance is irrelevant. We space it out for reading purposes. So I've given a lot up to this point. I haven't really paused. I've talked about my background. I've talked about the Listeria Initiative, our pilot project. some time at CDC. Do you guys have any questions at this point? Take a little break.
I'll ask a question, Lee. This is Katie. I don't think that you cover, like, does the length of the branch on the tree matter? Does that come into consideration?
Yeah, the length of the branch does matter. So this is so you can take your fingers and just measure it, I guess, or you can take a little ruler. But here's 0.01. So this is 1%. So 1% evolves on this branch length that much. 1% plus 1% plus 1%. So you can add that all up on this very large branch. I haven't measured up myself, but maybe you get 10% evolution right here. You get much less than 1% evolution on these very tiny, impossible to see branch lengths over here. Does that help answer the question?
Yes, definitely. Thank you.
Thanks for asking. All right, so back to the farmstead outbreak. This is the tree that we made at the time. Okay, we have this. So coded in red, blue, and green, we have red are the epidemiologically related clinical isolates. Those are the people who said that they ate the cheese, basically. Blue is the, I'm sorry, I got that a little bit backwards. Red are the cases that match the WGS profile. Blue are the cases that uh, happened before 2013. So it's not related to the outbreak. It's before the outbreak started, but they were found around the cheese plant. Green is the historical environmental isolates from the plant. So these are so after the outbreak happened, they went back and sampled the plant to find any listeria there, and they did find listeria there. So those are the ones from the plant. With a filled-in star, those are people who said that they ate the cheese. That's exposure. Those are the people who answered the epidemiologist that they ate it. Without filled in, that star, they did not eat the cheese. So we have pretty good concordance. The only reason that this star, I think in particular, this star is not filled in is because that patient had passed away, unfortunately, and the wife of that person didn't know if he ate the cheese. So what you can see here is a very large clade of low branch lengths, very small branch lengths, all related, and you can see clinical isolates related to environmental isolates. And that's sort of like a smoking gun. The same thing that someone got sick with was also found in the cheese plant. We also have other isolates out here, other taxa out here that are in blue. They are retrospective, not related to the outbreak. And that's just to help us root the tree and help us figure out the direction of evolution on this tree. We include that for the analysis purpose, not because they are epidemiologically relevant. So another fun tree to look at was in late 2013, a little bit after we started the pilot project. This was an outbreak that we didn't know the cause of. So we say we call it unknown etiology. We had about 10 people get sick from something, I think in North Carolina, but I could be wrong. Memory's getting a little fuzzy from that point now. And we had 4 cases that we sequenced right away and put into the tree. And then we compared it to everything else that we had at the time. We had very few isolates at the time. This was the farmstead cheese outbreak that I just described. This was, this much related. We also had retrospective isolates from 2009, a cold smoked whitefish recall from FDA, but that's from 2009, not related. And by the way, I might accidentally say recall versus outbreak. A recall is when no one got sick from it. FDA caught it before anyone got sick. An outbreak is when a few people get sick or more. So we had a question at this point. We still didn't know like how close was close. Like if something's like 2 SNPs different, is that epidemiologically related? If it's 50 SNPs different, we didn't know. So we put it all into a tree. Inside of this clade of people who we know got sick from the same thing, because it's just too coincidental. That was between 0 and 2 SNPs. The median was one high quality SNP. And then the difference between this outbreak and the whitefish recall is about 14 high quality SNPs. So is that related? Like, is it going to be like a whitefish related thing? We didn't really know yet. We also had related to it 10.5 SNPs away, a kale isolate and another isolate that we got from New York City about the same time. So we had some hints. Epidemiologists went in and asked these guys, and we're still trying to solve that problem, right? How close is close? So we started, the epidemiologist started asking, did you eat kale? And they're like, what is kale? So these guys are from a place in North Carolina that just doesn't know what kale is, and that's fine. Again, no judgment, but if they don't know what it is, they probably didn't eat it, and that's probably not the solution. They also answered that they all ate shrimp, and we're like, might be shrimp. Um, and we were about to announce that it was shrimp, except one person called back and said, "Oh, never mind, we never ate shrimp." And so that's the story of how we almost solved this one, but technically it is unsolved to this day. I see a question in the chat. Are transposons or plasmids involved in Listeria evolution? Sure. There are plasmids and transposons. I'll say up front that I'm not a huge expert on transposons or plasmids in Listeria. It has a large phage in it that can jump back and forth sometimes, and I believe it has a mechanism in that phage that inhibits future phages from plopping into there, so it can mark its spot. But other than that, I don't remember a lot of other mobile elements that have to do with the evolution of Listeria. Um, yeah, sorry, I have to say up front though that I don't remember a lot on plasmid or transposons themselves specifically on Listeria. Does that help answer the question? Oh, good question. All right, so I'll pause one more time if you guys have any other questions. Basically, Katie had so many questions for me to answer in this presentation. I was excited to answer all of them, so
I
I'm
have a
just
question.
glossing a little bit. Oh, sorry, go ahead.
Sorry, we're watching a party watching in the room here, and I had a
Nice.
question about the tree on a farmstead uh, outbreak. I don't know if is it possible to go back to that slide?
Yeah. To this slide?
Yeah, perfect. On the blue like 2012 L thing there.
Down here?
Yeah, I was just so that's closer to the other taxa than the other blue taxa, I guess. I'm not, I'm one of the people that need to learn about trees. So,
No shame. Okay. Yeah. Uh,
and just like, what does that
so
mean, I guess? Yeah, for like your analysis.
yeah. So this is an outgroup. It is not so related to over here. In fact, it's on its own clade, basically, if you can call it that. It's a singleton taxon. It's not related to anything really. You can kind of get an estimation of what it's more related to. You can say that it does share a common ancestor over here. But that's what, like 2%, 3% of its genome different than the common ancestor. And then you can add on to that this length over here, where that's different than the common ancestor. So you might get like 3 + 3 or 3 plus 2% different between this and the sister clade here. You can also make that kind of estimation between here and the nearest neighbor. Um, So it shares a common ancestor over here at this 100% confidence and this 100% confidence ancestor, and you can start making that addition over here. On the whole, just casually, I would say that it's just not related to anything else. It doesn't have anything with a short branch length next to it. Does that help?
Yeah, it does. Thank you.
Yeah, for sure. Thanks for asking.
I have a question.
Mhm.
Do you ever partner with, for example, with the listeria? Do they ever break this down in lab and see if there's any multi-drug resistant capabilities to this type of listeria, and then partner with them to see if there's any antibiotic resistant pieces to that, and then try and partner together on that with that research?
Yeah, and partnering is actually the right word here because I guess I'll say that we have a collaboration, and then you're asking about partnering, I guess, with academia, so I'll get into that too. So we actually have a very strong partnership with FDA and USDA in a big collaboration called NARMS. That's the National Antimicrobial Resistance Monitoring System. And our lab does regularly look at the antimicrobial resistance profile of each of these. For some, like salmonella, when we get too many of them, like there are a lot of cases of salmonella, they might look at like one out of every, I forgot what the number was. Maybe it's one out of every 20. Um, but they do look at these profiles. And then I think that you might have been hinting at collaboration with or partnering with academia or other entities. Sure, they have reached out in the past before, but that is unfortunately not my purview, so I can't really answer for that. Does that help?
Yes, thank you.
Sure. And you can look at the CDC website or the other agencies' websites for NARMS. They have some pretty awesome dashboards. You can download the data to it. Even if you're not partnering, I think you can do some pretty cool research just on the data you can download. Another part of this talk was Katie was just asking, I think these are great questions. Just what is the general career in bioinformatics for public health? So I've talked on this a couple of times, so fortunately I was able to grab a few slides on this. And so this is where I pretend to be an academic counselor for just like a couple seconds here. Because I don't know you guys, but I'm also rooting for you. Like I want the field of bioinformatics to be strong, and that starts with you. And that starts with you being honest with yourself and knowing what you want to do in life. So I'm hoping that everyone wants to go into public health, but the reality is that you guys have whatever plan that you have, and I hope that this all matches up in this comic. The plan that you tell your advisor, the real plan, and then the secret plan. So I hope this is totally in alignment. If it's not, then the point of this comic is be honest with yourself and know what you're doing. The other part of this slide is get yourself active online. I'm sure you guys are in some capacity or another, just because we're, in the year 2024. But that also includes making sure that you have your resume out there. If somebody contacts me ever about a job or even asking about this thing or that thing, I might search for that person, and you want the thing that you want to have control over your own search results. So make your LinkedIn the top result, or if that's not your thing, then make sure that your other profile online is your top thing, and you control your own narrative on there. Um, another fun slide is that there was this Quora article question: what is the day in the life of a bioinformatician? And somebody answered an amazing answer over here. And I guess I'll give these slides over to Katie, and she can send this out if you wanted to go through all this. Um, and by the way, I keep saying Katie, but also Jara, I know that you helped out a lot too, so thank you for getting me here. So I'll get us to Katie and Jara. Um, so it's just really funny. Wake up and prepare for the lab. go in and get your caffeine boost, kickstart, look at the computer for the last night's running programs. If it crashed, restart the program again. Like it can get just kind of tedious in these things, and then all of a sudden an outbreak happens and becomes exciting again. So this is the hilarious, tedious answer, but there are some times when you just get like an exciting thing. So that's sort of the day in the life. A typical career in bioinformatics and public health. You can go into a lot of different things. One of them is outbreak analysis. That's always exciting. I like to stay in applied research. So I showed you guys some exciting stuff. I do a lot of applied research, so comparative genomics and functional analysis. I like to look at those things. If if somebody asked me about plasmids and transposons in my lab, I would have been all over it. That's the kind of stuff I like to nerd out to. I like to work on pipeline which is workflows development, making a system that takes your Illumina reads and turns it into some knowledge, some results. A typical career is also very collaborative. In fact, I try to mirror that in my own Georgia State course. So in my Georgia State course, I tell them all up front that this is open book, open internet. You need to ask your friends, start a group.me chat between each other. I don't need to join it. Just talk between each other. And that's been working out a lot because the reality is that when you get to your career, it's highly collaborative. You have just graduated and you don't know everything. And even 15 years into your career, you don't know everything. So you always need to ask people things. And some of that collaboration might be just talking to your teammates. You might have collaborators. So I appreciate that earlier question about partnerships. Training, there's lots of training. And then going to conferences and learning from people abroad. And then some of your work might be the more boring stuff like system administration, purchasing, IT advice, somebody walking up to you and saying, "Can you fix my printer?" Hopefully you can avoid that. Bioinformatics at CDC. Bioinformatics at CDC, I want more people just personally at CDC. In 2015, we had fewer than 10 people who identified as bioinformaticians. After 2020, the last time that this was counted, we had more than 75. I personally believe we probably have over 100 or 200 people now in bioinformatics strictly. I'd like that to be more, honestly. So that's why it's one of the reasons why I love talking with students. And maybe just for the sake of time, I'm going to zoom ahead. But we do have workforce capacity and lots of training in CDC. There's lots of ongoing training, and some of that is stuff that I help lead. I like showing this slide because, especially here today, do you recognize anybody, Katie?
I think the center picture is Milan, but I could be wrong. I also posed for
Isn't
one of these pictures, but I'm not in any of these, thank goodness.
wasn't this one you?
No,
That one
that was
wasn't
a
you?
different
Okay.
one. No.
All right. Katie is one of these, but I guess I didn't show this one today. Yeah, this is Milan and Grant over here. They produced these posters for conferences. This is the Office of Advanced Molecular Detection, OAMD, and they help organize genomics at CDC, and they're sort of like our center, our hub for genomics and bioinformatics. To get into CDC, there are a few different mechanisms. One of the more popular ones is the Oak Ridge National Lab Fellowship. And I always get a question, so I'm just answering this ahead of time that yes, this is a mechanism that can take non-citizens. They have a strict policy, a rule that you have to have graduated within the last 5 years. So we post jobs there a lot. This is a good website to look at. And then for citizens, the APHL Bioinformatics Fellowship is amazing too. This is normally for people who just got their PhD or who are just about to get their PhD. And you get placed in a lab for 1 or 2 years to work on a project, and it's a good way to get your foot in the door. The Bioinformatics Fellows have been wildly successful, and so this is a chart of our retention for Bioinformatics Fellows. I got to have one of the first of the 3 Bioinformatics Fellows in 2013. She stayed with us. And let me see. Roughly 70% of the graduates have stayed on at CDC or in the state or local levels in their roles. This is still a very strong fellowship and highly regarded. We also have a lot of next generation sequencing capacity in public health laboratories. This is an older slide for sure. So I'll just click through that. So over time, we do have capacity for sequencing genomes in all the state health labs. I know I'm coming from CDC, but partnerships are extremely important. We cannot look over every single thing happening across the whole United States. So this is to say that number one, we need the state health labs, and number two, yes, you might have heard of CDC, but have you also heard of the Georgia Department of Public Health? So there are lots of places locally that you can look at careers, lots of places. And there's a growing list of applications for next generation sequencing, including all of these pathogens. So there's lots to study, lots to address. And if, for example, Florida is an amazing state to you, then you can start looking at the Florida State Department of Health website for any jobs that they might have, or whatever state that you like. And there's bioinformatics beyond CDC, of course. I think about 5 or so years ago, we compiled this list of different careers websites. These are some places where you might find bioinformatics at CDC or otherwise. And I'll show you a link in a few more slides where I compiled this. There are also careers in the federal government. So the main site for that is USA Jobs. You might have noticed I showed you fellowships at first, and that's because you can get placed inside of CDC, but not be employed directly by CDC. And then this is the place where you would get employed by CDC at usajobs.gov. We also hire from contractors. We hire contracting companies. So you could work for a contracting company. That government would pay money to that third-party company to staff somebody at CDC. So that's a government contractor. I went to Wikipedia just to get an idea of who these contracting companies are, and there is a Wikipedia article of the top 100 contractors of the US government. So you can take a look at these guys here. There's also careers in industry itself. So there's third-party testing labs in food companies. The Center for Food Safety in Griffin from UGA, they deal a lot with these companies that are looking at food. They hire bioinformaticians. For example, like you might not have even guessed, like Nestle has a bioinformatics team. So you can look at some food companies and see if they are hiring also. Some have adopted sequencing technologies and have a need for bioinformatics expertise. So you might also look at hospitals or nonprofit organizations. These guys are hiring too. Anything analyzing health-related data, they are hiring bioinformaticians. You also have biotech companies, big pharma. There's software development out there. You might take the algorithms that you learn in bioinformatics and just go completely software. That's a thing. And yeah, that's competitive. It might require, higher level degrees or expert knowledge, but it's a thing you can do. There's also careers in academia. Academia is great. I really like that. That you can be a bioinformatics staff. You can go to a university sequencing core, for example. There's staff scientists. Okay, I looked at the clock one more time. I'm going to just zoom ahead, but you'll have these slides. These are all ideas. And yeah, here's the Center for Food Safety. This is the website where you can see my group that I'm adjunct with. And they are definitely looking for people who are more interested in food who are coming out of the bioinformatics area. So additional resources. I got my slides from a talk that we did, I think in 2019 at this URL, this Google Drive. I compiled the resources for those jobs here at this website. Some people also ask me, how do I level up? How do I learn Python? How do I learn RNA-seq? Like whatever. I started compiling a list of different ways you can get educated on your own. So you can go to this website also. All right, so the bioinformatics definitely is getting popular. This is my class enrollment over the last few semesters. So it's gone from, I think I started at 20 students and I'm up to 35 this semester, and I'm waiting to see what next semester's enrollment is going to be. So I'm delighted by that. I'm also on social media, especially in the podcast space. So I also did like a little survey. If you're somebody like me who just likes to listen passively to someone talking about a topic you'd like to know more about, I compiled a few podcasts and YouTube channels for that. I really like these. This one's my own over here. This is my colleagues, the Bioinformatics Lab. There's this guy who put like his whole bioinformatics course on YouTube, and he's really good. This is a graduate, an alum from my own lab at CDC. She went on to become bioinformatician, and she has a great YouTube channel. Um, this person, Georgia, she's really great. I like how Georgia explains things as a person in her first year in her career. She's really good at it. And there's the OM Genomics podcast. These guys who have gone out on their own independently to create their own company, and they talk about things from their perspective. They're really great too. So my own podcast, I'm the co-host. I'm one of the co-hosts with my friends Andrew and Nabil. Um, you can listen wherever you listen to podcasts, and we did it because we realized that bioinformatics is so rapidly changing. We needed a place to honestly to vent to each other and just get our bioinformatics conversations out to somebody who wants to listen to us. So we basically talk to each other. Uh, but we just decided to like have a little bit more fun with it. We on our first year anniversary, we had how we made the podcast. But more recently, I think last December, we loaded up Jurassic Park and watched it together and just commented on the bioinformatics of Jurassic Park. We're thinking about doing something like Gattaca or Contagion this year. We haven't decided yet, but we think we're gonna make that a thing. So come listen if you want, and if you don't, that's okay too. It's really for my own personal mental health to talk with my friends here. So with that, if you have any questions, I'm on all these websites. You can come find me. You can email me, and I'll open the floor for any more questions.
Thank you so much, Lee. I love this presentation. I've definitely perused a lot of your GitHub for especially like the employment options, because it's good to have an idea of what to get into. Also, not to brag, but to convince myself that I'm working during times that I'm not working, I listen to your podcast while I'm crocheting because it feels like I'm still doing bioinformatics. So thank you for that.
For sure.
So
That's
I
hilarious.
appreciate it. Yes, if anyone has any questions, please unmute yourself or write something in the chat. And for those that are in the Davison classroom, I'll leave it up to Olivia to let me know if there's any questions there.
Hi, great talk. I wanted to ask about how the kind of project directions get decided on at the CDC, because how I'm used to it is, it's about the interests of the PI or of the student, and then also what can get funded in a grant. But I was just kind of curious how that kind of process works for you.
I mean, optimally, any project starts with a need, or proactive need. Sometimes I see something that needs to get done before it's too late. So I'll go out and do it before someone asks me. But usually, something will happen because of a need. So with the Listeria pilot project, for example, we just got our sequencer and they put 4 people on making a protocol on how to sequence DNA and how to QC, how to do quality control on your reads. Um, they put 4 people on the wet lab side basically, and then they said, as an afterthought, they were just like, "Can you figure out the data management for that?" And I was like, "Sure." So this actually turned into a whole project, which I finally published a few years ago, called SneakerNet, and it was how to move the reads. We had to do it on a hard drive, so we called it SneakerNet. You walk the reads from the sequencer over to the computer for storage and further QC. Now we don't have to use the hard drives, but it's still called SneakerNet, and it's a whole process on how to place the reads in the right place in our local repository, how to QC the reads and write a report, an automated report to everybody who wanted to receive the report. And that's just a case example of a need that turned into a project. Other times, and then maybe one other way of looking at it is who comes up with that need. For whatever reason, nobody usually argues with me when I just say I want to do a project. But for most people, like they would say, I think that I should do this, and they would probably go to their supervisor, have a little discussion, and then they turn into an official project. In the last couple of years, people have added project innovation into the bureaucracy. So now you also write up a little form. Um, if that's your cup of tea, then go for it. You can write up that little form. Um, but basically, like I would say the people who are brainstorming come up with the ideas, and then if you have a good supervisor, then they listen to you and you go out and do it. Um, did that kind of address it, or I kind of feel like there might
Yeah.
be one more place that you asked the question that I didn't address.
No, that's pretty much exactly what I was asking. That's a lot more independence than I guess I was kind of expecting. But yeah, thank you.
Oh, uh, okay, you're welcome. And I have one more answer for you. Sorry. So this was you didn't ask this part, but this was in the back of my brain while I was talking. For fellowships specifically, while you're applying for the APHL Bioinformatics Fellowship or the ORISE, you might be asked to come up with a project idea as part of your application. So in that case, the project idea might actually come from you. And then there's a matching system with the host lab, and they prioritize which projects look best for them. And then there's that matching program. So you might have already come up with the project as you applied for it too. I appreciate that question. Thanks.
We have a question in the conference room.
Go ahead.
One last super quick question. So when you're doing new projects and you have to program in new elements to facilitate the project, especially when you're collaborating with the FDA or anything to that nature, do you have to run validations on the programs? Who helps you with that? Is that something you have to do? You know, the FDA is obsessive about their validation summary reports, and how does that
Yeah.
work? Do you people in your department to do that, or do you just? I'm fascinated.
Yeah. Yeah, validation is a huge thorn in my side sometimes, but it's a necessity.
Yep.
You have to do it. And I'm actually surprised sometimes when people don't do it.
Yeah.
Um, I, I have have so many thoughts on this. So if it looks like I'm pausing, just trying to think of where to start on this. So you have to do it. Like if you're going to have a process that affects public health, you need to validate. And usually that starts with a protocol. So can your protocol get validated? And your protocol might be, this is how I pass or I fail my reads on my quality control pipeline. So I'll stay with that same software because I've already talked about it. How do I tell the people in the sequencing lab that their reads have passed or not? What is pass or fail? So we might say that Q30 is a pass, but then we need to validate it. So then the other place that it starts is a benchmark data set. And I have gotten on my soapbox so many times for this actually that I do have a repo in my GitHub here for benchmark data sets. PulseNet puts out benchmark data sets, other places put them out. I was surprised during the SARS-CoV-2, during the COVID outbreak, nobody had benchmark data sets out yet, and I went ahead and jumped on that and I published those too. And people use my benchmark data sets now to publish, and to validate. And you can validate on anything. I said QC, but in my validation data sets, you can have things like, here's what contamination looks like. Does your QC pipeline pick up on contamination? Does it pick up on low sequencing coverage? So you can put out deliberate errors in your validation data sets too. In the realm of just software development also, there's this idea of continuous integration, and I've published on this as well. I'll try to remember to follow up when I send this presentation with that URL. When you're doing that, you actually need, when you're developing bioinformatics software, you actually need to develop automated tests, in my opinion, and that helps you in not only in the software development to make sure your software keeps running every time you change it, but also to show you that you get the right result every time you run it. So if you've made a pipeline that detects contamination, or let's say it detects serogroups or transposons, like does your software detect the same transposon every single time? Or after your last modification, did it fail to do that? You need to have that continuous integration. So I've published on that with suggestions on how to validate your software also.
All right, thank you so much, Lee. Unfortunately, we are out of time, but I do want to thank you again for spending the past hour with us. And it sounds like you're comfortable with me sharing your presentation, so I'll do that as well. And yeah, again, thank you so much. It's good to see you again.
Thanks for having me. I really appreciate it. And I'll send you the updated presentation. I've changed a few slides since then.
Okay, thank you. All right, everyone have a nice weekend.